Okay, so today we are going to do adenovirus. The adenovirus is the virus which is icosahedral, non-enveloped, double-stranded DNA virus, and it belongs to Adinoviridae family. And there are two subtypes or two genera: AV adenovirus and masked adenovirus. Now there are 7 species from A to G. From A to G, A, B, C, D, E, F, G till G, and the B1 has two subtypes, B1 and B2. Now it has 252 capsomeres, and as I have told in the morphology, it is icosahedral. Capsid is present at containing double-stranded DNA. Now in the structure, suppose this is the structure, icosahedral structure of the virus. Let me make it first. I am just making a rough diagram to give you an idea. So please don't mind about the drawing. So now some projections like this are coming out of the virus. So these are phanton having a fibrous stalk and a knob which binds to specific receptor sites on the host cell. Now there is let's talk about the resistance of this virus. The virus can be active in 37 degrees Celsius for weeks. It is resistant to ether and bile salts. For the pathogenesis, it affects respiratory tract, conjunctiva, small intestine, urinary tract, basically GIT. As I've said, small intestine, so it affects GIT. So the primary infection is in the conjunctiva or the pharynx or the small intestines. From there, after multiplying, it goes or it spreads into the lymph nodes or drain into the lymph nodes, and the incubation period is 5 to 7 days. So let us talk about the immunity. Maternal antibodies provide immunity to the child, and once the infection has occurred, there is long-lasting immunity. Now for the laboratory diagnosis, first we have specimens. So as you know, the pathogenesis or diseases which occur, so what can be the specimens? It can be swab from the conjunctiva or the throat swab or the nasopharyngeal aspirate, urine, and also the genital secretion biopsy. So what do you find or what do you do in direct microscopy? You can directly check the virus particles in the feces because it spreads through conjunctiva, nasal, mucosa, fecal spread is also there of it. And you can take the scraping of cornea also, and from here you can demonstrate the virus through immunofluorescence using monoclonal antibodies. Now isolation from these cultures or from these areas like throat, urine, feces, the virus may be isolated in the human embryo kidney cells or HeLa cells or cell line or Hep2 or KB. Now what else you can do in laboratory diagnosis? Latex agglutination test, you can do PCR, you can do serology, rise in titer of antibodies which can be demonstrated, and I told you that these don't have capsid. So basically, if the virus doesn't have a capsid, it is difficult to kill that virus, and we don't have vaccines for it because it has a large number of serotypes and a large number of subtypes. So that's all about the adenovirus. Please like, share and subscribe the channel.